Serotonin is a neuromodulator. And when we think about the function of serotonin, it particularly exemplifies this idea of modulating behavior. A quote that I really like from a paper from a couple years ago is that serotonin is involved in everything but responsible for nothing. And this is because there are so many behaviors where if we looked, we could find that serotonin does influence that behavior and that it modulates it, pushes it more in one direction or another. But there really isn't anything that's been identified that serotonin is solely responsible for. So this quote of serotonin is involved in everything but responsible for nothing really exemplifies how modulatory serotonin is in its function. We can see how important serotonin is in different brain regions by looking at the anatomy and the pattern of projections of serotonergic neurons. Much like the other monoamines, serotonin is produced by neurons that are located in the brainstem. So we see these discrete clusters of neurons in the brainstem, in the pons, and in the medulla that house these serotonin neurons. Now remember, the catecholamines were the A cell groups. Serotonin is labeled as the B cell groups, which you see are kind of dispersed throughout the brainstem here. These B cell groups are loosely affiliated with what are known as the raphe nuclei in the brain. And the dorsal raphe nucleus is the largest and best understood of the serotonin nuclei in the brainstem. So the dorsal raphe is located in the brainstem, and it contains serotonin neurons, which, as you can see, project throughout the brain. And in fact, there really is no region in the forebrain or in the limbic areas of the brain that doesn't receive serotonin's innervation. So this is a neurotransmitter that is widely distributed and influencing all types of brain regions and the behaviors associated with those regions. Now this figure here shows us the projections to the dorsal raphe, and we can also get a sense for serotonin's function by looking at what projects to it. So we see neurons in the brainstem associated with some of those actually acetylcholine neurons associated with arousal project to the dorsal raphe. We also see neurons in the hypothalamus, the midbrain dopamine-containing regions of the brain, the substantia nigra and VTA, project to the dorsal raphe. An area of the brain called the lateral habenula, which we're learning is involved in aversion and mediating negative emotion, projects here, as well as the prefrontal cortex. If serotonin is responsible for nothing, then what does it do? The best characterization I've seen of the function of serotonin is that it mediates coping, and that means coping with stress. So just as dopamine could be quickly summed up as mediating motivation and norepinephrine seems to mediate arousal, we can think of serotonin as mediating stress coping. And this is, I think, a very nice synthesis of the different functions that we see serotonin mediating. And if we look again at the projections, we can see that this makes sense as well, that serotonin is getting information from regions of the brain that have to deal with motivation and negative emotion, as well as top-down control. So these brain regions are activating serotonin and then allowing it to signal during times of stress to help the rest of the brain deal with that stress. And we'll walk through some examples of exactly how serotonin accomplishes this function by looking at some specific behaviors that are influenced by serotonin signaling. Let's start out by thinking about serotonin's role in the sleep-wake cycle. Now, I made the argument that norepinephrine and cholinergic activity as well is critical for arousal. We also see that serotonin signaling seems to be related to the sleep-wake cycles. So what you can see here is the firing rate of serotonergic neurons in the DRN, and that they're most active, or they're firing the most during periods of active waking. So when animals are exploring and actively engaged with the environment. They slow down when the animals are in more of a quiet waking state, and then are almost silent during slow wave sleep and completely silent during REM sleep. Now, we could from this conclude that serotonin must be involved in producing a state of wakefulness. But what we actually know is that the DRN receives projections from the cholinergic neurons in the brainstem as well as from the locus coeruleus. And so it seems to be the case that rather than serotonin producing wakefulness, that serotonin activity is a product of wakefulness. That when these other systems start to wake up the brain and turn the brain on and bring about that state of arousal, they're also turning on serotonin signaling. And so if we think about serotonin as mediating coping, what this tells us is that during sleep, when we're not getting any sensory input and we are presumably not very stressed, serotonin isn't needed, and serotonin neurons are quiet. But during waking, and especially when we're exploring and actively engaged with the environment, this is when we get the most serotonin signaling. This figure from your textbook sums up a variety of functions of serotonin by summarizing what happens in serotonin transporter knockout mice. So these mice don't have SERT, which means they can't remove serotonin from the synapse, and we would expect them to therefore have chronically high levels of serotonin. Now, because they have these high levels of serotonin throughout their life, they're actually going to have a variety of other adaptations to those high levels of serotonin. So the direction of the behaviors here, whether these things are increased or decreased, isn't so important. But what is important to pay attention to here is in these SERT mice where you have chronic dysregulation of serotonin signaling, we see changes in susceptibility to stress, to anxiety, in sleep, depressive-like behaviors, and aggression. The rest of these things over here are peripheral effects of dysregulating that serotonin signaling, which tells us that serotonin receptors are expressed in the periphery, and they are. But we're more interested in what's going on in the brain and in the behavioral functions of serotonin. And all of these things, anxiety, stress, depressive behaviors, aggression, all have to do with that coping behavior. Serotonin is involved in reducing anxiety, helping us deal with stress, elevating mood, reducing aggression, as well as impulsive behaviors. So the SERT knockout mouse actually gives a lot of clues to the functions of serotonin, and we'll dive into some of these more specifically right now. Another knockout mouse that's been created is the TPH2 knockout mouse. So this is a mouse that is lacking tryptophan hydroxylase. This mouse cannot synthesize serotonin because it can't convert tryptophan. And so we see a near complete loss of brain serotonin in these mice, and that's what this graph is demonstrating here. These are our wild-type control mice that have 2 functional copies of the TPH2 gene, and they have 100% normal levels of serotonin. Mice without the TPH2 gene have almost 0, right, percent of the control level of serotonin. So they have this complete loss of serotonin in the brain, and we can actually increase some of this serotonin and bring back that serotonin synthesis by giving 5-HTP, which remember is the next precursor to serotonin that normally tryptophan is converted into by TPH. And so by sort of bypassing TPH and giving 5-HTP directly, you can still get synthesis of 5-HT, and we can restore some of that serotonin signaling. So these mice have also been helpful in understanding what serotonin does. And if we look at for example, the behavior of these mice in a test of aggression, we can see how serotonin is involved in reducing aggression. So we're looking here at an intruder test. So animals are, this is usually done in male mice, which tend to be aggressive toward each other, where two stranger mice are put together. And we can look at parameters such as the latency to attack the other mouse and the number of attacks that are done. And you can see in wild-type control animals that they have a longer latency to attack than the knockout mice, which don't have serotonin in their brain. And they also make fewer attacks than the knockout mice. So mice without meaningful levels of serotonin in the brain are much more aggressive than their control counterparts. And we see that again here in this graph, which also demonstrates that if what happens if we restore some serotonin by giving 5-HTP, and we can see here that increasing the levels of serotonin in the brain with 5-HTP will decrease the latency to attack and reduce the number of attacks. So here again, by increasing serotonin, we've made animals less aggressive. So this is one example looking specifically at aggressive behavior, which shows us that serotonin is involved in decreasing aggression. We also see that serotonin is very important in anxiety-like behavior. This study was done specifically by looking at the 5-HT1A receptor. Remember, this is a G protein coupled receptor that has an inhibitory influence postsynaptically. And what we can see in an elevated plus maze is that animals with knockout of 5-HT1A, so they don't express this receptor, will show less time in the open arms of an elevated plus maze and less distance traveled in those open arms. This is indicative of anxiety-like behavior, so they're less willing to go out into a scary place and explore it if they don't have 5-HT1A. This tells us that serotonin signaling through this receptor is important in reducing anxiety-like behavior in a stressful situation. All of these functions together really have been summarized as serotonin's role in coping. And there has been a more recent hypothesis thinking about 5-HT1A and 5-HT2A as mediating passive coping and active coping, respectively. Passive coping would be helping deal with stress without actually making any response to change that stressor. And so as we saw in the elevated plus maze study, 5-HT1A signaling can decrease our feelings of anxiety. It can decrease feelings of stress and aggressive behaviors, and also can make us less impulsive. So more likely to wait and be able to sort of make more measured decisions when we have proper levels of 5-HT1A receptor signaling. Overall, we can think of this as being more resilient or more patient and more tolerant of stress. 5-HT2A is involved in things that have to do with active coping. And so this is actually making a behavioral response to diminish the stressor that's in your environment. And 5-HT2A seems to be much more important in this. And it therefore shows up in behaviors like flexibility and negative thinking. And so we see that 5-HT2A is actually making us more flexible and able to make decisions that sort of help us deal with our environment. And so we think of it as being involved in adapting, and learning to deal with what's going on.